Hello everyone. Today's topic is about plasma membrane key differences in the Archaebacteria and in bacteria. Many a times question arises from this topic regarding the key differences in the plasma membrane composition in these two organisms. So we will be talking about these differences one by one. There are basically four differences. I will be talking about them and will be telling you the trick how to remember these. Okay. So the first is plasma membrane in both is composed mainly of lipid, glycerol and phosphate. And we know that glycerol exists in two forms, mainly L form and D form. Now the question arises, in which organism does L form exist and in which does the D form exist? So the trick for this is B and D letter somewhat looks alike. Okay, so in bacteria D-glycerol is present and in Archaebacteria L-glycerol is present. Second key difference is linkage between the lipid and the glycerol. In which organism ester linkage is present and in which the ether linkage is present. Now the trick is, in Archaebacteria the letter H is present and in ether also letter H is present. So in Archaebacteria ether linkage is present between lipid and glycerol and in bacteria ester linkage is present between lipid and glycerol. Okay. Now the third key difference is whether the phospholipid is bilayer or monolayer. The trick for this is, as we know that Archaebacteria live in extreme harsh conditions, so it can be thought like this, that when someone is in harsh stressed condition, the individual wants to get united, isn't it? So there is a Hindi line for this, एकता में बल है, चार लोग साथ रहेंगे तो हिम्मत आएगी. Now here ekta represents monolayer and char represents tetraether. So in Archaebacteria, tetraether monolayer phospholipid is present and in bacteria normal phospholipid bilayer like that of eukaryotes is present. The last and the fourth key difference is whether the isoprenoid lipid chain is branched or it is unbranched. To remember this, there is a trick that, as we know that Archaebacteria survives in extreme conditions, so we can say that they are a bit complicated. No, they are not simple, and as branches represent complexity, unlike linear simple form. So in Archaebacteria, branched isoprenoid lipid chain present, and in bacteria unbranched or linear chain is present.